Beneath the surface of pond, thousands of prawns are growing silently and generating huge income. Would you believe that these small aquatic creatures can turn a simple pond into a profitable enterprise? What if a single pond could become your seafood factory? Welcome to the world of prawn culture. From hatchery to harvest, watch how science and nature come together to produce premium quality prawns. Top of form. To get maximum information, watch complete video without skipping any part. To get maximum information, watch this video till end without skipping any part. In this video we are going to study hatchery system, spawning and larval rearing, grow-out techniques, food and feeding, disease management, harvesting and marketing. Let's see how spawning occurs under optimal conditions. Prawns can spawn in captivity. Buried, gravid females are collected from commercial harvest. For brood stock selection, individuals above 4 to 5 grams in weight are preferred as the quantity of eggs spawned is comparatively high. Spawning done in fresh or slightly brackish water which has salinity range from 2 to 8 ppt. These brooder females should be fed on mussels, cockles, fresh trash fish or compounded feeds. Stocking density can be maintained at a rate of about 1 to 500 hectare. Weight of gravid female should be near 200 gram. The eggs change in color from orange to grayish-brown as they mature. Mature male prawns are considerably larger than the females. Second walking legs of male much larger and thicker. The cephalothorax proportionately larger. The abdomen narrower. The male can also be distinguished by the presence of a lump or hard point in the center on the ventral side of the first segment of the abdomen. Hatchery system. There are various hatchery systems in use in experimental centers and commercial farms, but the more important ones can be described as belonging to the green water, clear water, and recirculation systems. All procedure in same tank. In many hatcheries, mating, spawning and larval rearing occur in the same tank. Larger tanks are required for water storage, post-larval rearing and for hatching artemia. The tanks may be in the open in tropical areas, but are usually kept under a roof to prevent the water from getting too warm during summer days and too cold at nights. In temperature climates, it is necessary to house the tanks in a greenhouse-type construction to maintain the water temperature at the required level during the cold season. Let's see which types of tanks are best for this purpose. Tanks to be used in prawn culture can be made up of plastic, fiberglass or reinforced concrete. Circular tanks with flat or conical bottom, easy to clean and better water circulation can be maintained. Rectangular tank are most convenient and space-saving. A 10-meter cubes tank, with water and air intakes at one end and the drain at the other, has been found to work very efficiently, convenient and space-saving. Square-shaped tanks. Tanks to be painted with a dark color. Better feed visibility for larvae. Aeration system at the bottom of the tank helps proper mixing of the water and even distribution of the food particles and the larvae, thereby reducing cannibalism. In the presence of stacked solid layers, the larvae exhibit what is termed an edge effect, which is a pronounced preference for the larger edges. Instead of spreading out evenly across a flat surface, larvae tend to cluster along the edges. The units consist of a rigid frame to which layers of plastic mesh are attached. The mesh layers increase the area of surface edges significantly, both vertically and horizontally. Strips of screening placed on different layers provide feeding spots where the food settles. By providing significantly more edge space than a flat surface would, these units allow for a much higher concentration of larvae to thrive in the same amount of physical space as shown in the following figure. Water quality parameters for prawn. Bore-wells water provides unpolluted fresh and salt water. Water pH must range between 7 to 8.5. Water temperature be kept between 28 to 31 degrees centigrade. Nitrite should not be higher than 0.1 ppm. Nitrate levels should not exceed 20 ppm. For prawn culture, 3 types of water can be used: green water, clear water and recirculating water. The green water system, as the name implies, involves the production of a mixed phytoplankton culture dominated by chlorella at a cell density of 7500001500 cells ml. A solution of a fertilizer mixture of 4 parts urea to 1 part NPK, 15-15-15, in tap water is added to the tank at least once a week to maintain the plankton bloom. Rotifer's production in the tank is controlled by using copper sulfate at a rate of 0.6 ppm. Filamentous algae growth controlled by holding T. Mosambica in the tanks at a rate of 1 fish per 400 liter. Only green water less than 3 days old is used. For a continuous production of such water, without causing phytoplankton crashes, it is diluted regularly and fertilized as required. Function of the green water is to maintain the water quality in the larval tanks, recycling the waste products of larval metabolism and provision of food for live food organisms such as Artemia. The clear water system can be adopted for different levels of larval production. Circular tanks with conical bottoms or rectangular tanks are used. The system developed by Aquaculture for high-density larval culture involves the use of conical bottom tanks of 800 liters capacity with total water exchange and environmental control, including temperature and light intensity. Water quality maintenance by chlorination, followed by UV light, storm aeration and the use of antibiotics. Stocking density 1 to 200 larvae L yielding 25 to 100 post larvae L are feasible, but require very careful management. Conservation of water and energy. The simplest type of this system recirculates the tank water through a graded sand gravel filter using mechanical or air lift pumps. More complicated, completely closed systems involving mechanical and biological filtration, chlorination and dechlorination or the use of ozone have also been developed. Though the value of such a system in controlling the water quality is recognized and is utilized in experimental work, commercial application on any significant scale has yet to be realized. On the other hand, efforts are made in commercial hatcheries to operate both green water and clear water systems with only limited water exchange. Spawning and larval rearing. Pre-mating molting. Mating, deposition of semen, extrusion of eggs through gonopore, fertilization of eggs, transfer of fertilized eggs to brood chamber, located under the abdomen between the pleopods. It is believed that an enzymatic reaction is involved in the release of sperm cells from the spermatophore. The fertilized eggs become attached to each other and the setae of the first 4 pleopods by a cementing substance which hardens into an attachment membrane. Mating takes place between hard-shelled males and ripe soft-shelled females which have completed their premating molt. Semen is deposited in a gelatinous mass on the underside of the thoracic region of the female, between the walking legs. Within a few hours, the female extrudes eggs through the gonopore and the eggs are fertilized by the semen attached to the abdomen and transferred to a brood chamber. The vigorous movements of the pleopods keep the eggs well aerated. The number of eggs laid varies with the size of the prawn, but it is reported to be up to 80,000 to 1 lakh per spawn. However, a 1-year-old female may spawn only 5 to 20,000 eggs during its first spawning. The incubation period ranges from 18 to 23 days at temperatures of about 28 degrees centigrade. Even during the incubation period of fertilized eggs, ovarian eggs start maturing and a second premating molt can occur within a period of about 3 weeks. For spawning purpose, healthy buried females can be taken from natural resources, rearing ponds, brood tanks or aquaria. In community rearing tanks, it is necessary to ensure that all of them have black or grey eggs, so that they will all hatch out within 2 or 3 days and the larvae in the tanks will be of the same age. This helps in appropriate feeding and reducing cannibalism among the larvae. Physicochemical conditions during spawning. Salinity of water in hatching tank should be managed at 5 ppt. Salinity of the water after hatching should be 12 ppt. Optimum temperature range must be set between 26 to 31 degrees centigrade. Aeration in the tanks should be maintained in order to maintain oxygen levels. Avoid direct sunlight, instead providing indirect light. Removal of spent female from hatching tank over a 4-day period with a peak between 24 to 72 hours. Brackish water with a salinity of about 5 ppt is provided in the tanks as hatching rates are lower in fresh water. Water of the required salinity is obtained from mixing tanks in the hatchery. Larval stages. Macrobrachium undergoes various stages during larval development. Time taken for a batch of larvae to metamorphose varies according to temperature and feeding conditions. Under favorable conditions, the post-larval stage is reached in 16 to 28 days. The past larvae resemble juvenile prawns and rest or crawl on the tank surfaces. At this stage they are harvested from the tanks for nursing or on-growing. As shown in the picture. Stocking density in larval tanks. Start with a higher density of 80 to 120 per liter of water. A final density of 40 per liter is recommended. In small rearing ponds, it may be possible to carry out the rearing without any division of stock. In hatcheries following the green water system, water in the tanks is lowered daily to a 50% level and replaced with green water. In clean water systems, after removal of about 50% of the water, clean water is used for refilling. Feeding of larvae, fish flesh, fish roe, egg custard, egg and mussel mixtures, compound feed. As macrobrachium larvae do not actively search for food, feed the swimming nauplii of Artemia twice a day till 5th day after hatching. Prepared feeds are generally given only from the 5th day, even though weaning can be started a couple of days earlier. These are given 4 or 5 times during the day, and it is recommended that the night feeding should be with Artemia nauplii only. The quantity of prepared feed is also determined by the need for a suspension of it in the tank to make it easy for the larvae to locate it. The quantity needed at each feeding has been calculated to be about 30 to 60 gram per tank initially, gradually increasing to 200 gram. Vigorous aeration helps to keep the feed in suspension near the larvae. As mentioned earlier, the time taken for a larval batch to finish metamorphosis depends on the environmental conditions, but it is usually completed in less than 28 days and it is best to harvest them at this stage. The post-larvae can then be transferred to fresh water, but it is advisable to acclimatize them through a gradual lowering of the salinity by draining part of the brackish water and replacing it with fresh water. Production of larvae. Between 1 to 2 lakh larvae from a 10-meter cube tank. Production ponds. The post-larvae are stocked directly in production ponds or grown for a period of 1 or 2 months before on-growing to market size. Size of production grow-out ponds must be between 0.2 to 0.6 hectares. Depth of production grow-out pond can be adjusted up to 0.9 meters. Water salinity, fresh slightly brackish, must be less than 4 ppt. Optimum temperature for high growth should range between 26 and 30 degrees centigrade. Water quality management is generally through water exchange, though paddle wheels or other aeration devices are maintained for use in emergencies. Stocking rate depends on preferred market size, length of the growing season. The most common grow-out practice has been described as the continuous culture or continuous stocking and harvesting system, in which the ponds are stocked once or several times a year and are never drained except for repairs. Harvesting. Harvesting is carried out selectively for marketable prawns about 30 to 45 gram size at regular intervals. A considerable disparity in growth rates between individual prawns, especially between males and females, occurs in ponds. So selective fishing is performed as a means of stock management and to grow undersized individuals from previous stockings to marketable size. If the whole stock is harvested together, there will be a number of small prawns which may not be acceptable in the market. Prawn ponds following this system in Hawaii are reported to produce about 276 gram per month by selective harvesting, yielding about 3,314 kilograms per ha per year. Some farms in Hawaii have started incorporating a nursery phase or an intermediate grow-out phase to avoid some of the problems of continuous stocking. This requires specially constructed ponds with harvest sumps. Food and feeding. The types of feed used in freshwater prawn farming vary considerably, from locally available feedstuffs to compound chicken feeds and specially formulated prawn feeds. Dietary protein requirement, 20 to 25% or maybe 40%. Feed ingredients used by Asian farmers, rice byproducts, trash fish, mollusks, prawn waste, poultry waste, meal of ipilipi leaves, feeding rate, adjusted according to assessments of daily consumption and biomass changes. Recommended rates, 2.7 to 7.4 kg per hectare per day in the early rearing period, 37.5 kg per ha per day towards the end of the rearing. Feed conversion ratios generally range from 2:1 to 4:1 for commercial diets, giving growth rates averaging around 1 to cm month. Polyculture. Since M. rosenbergi is a benthic omnivore, it is considered a suitable candidate for polyculture. Experiments in polyculture with Tilapia aureus did not reveal any significant interaction between the species. Relatively high combined yields were obtained in polyculture trials with tilapia, channel catfish and Chinese carp in Alabama. However, The yields and individual harvest weights of the prawns seem to indicate interspecific competition. Disease outbreak. Reasons include poor sanitation, inadequate water exchange, poor feeds and low oxygen levels. Disease caused by epibionts. The epibionts are mainly filamentous and non-filamentous bacteria, algae or aquatic protozoa. The protozoan agents include the genera Epistylis, Zoothamnium and Vorticella. These organisms attach themselves to the epicuticular surfaces but do not cause any inflammatory responses. Zoothamnium appears to attach itself to the gill lamellae, while other protozoans do not exhibit any site specificity. Bacterial fouling of proximal appendages, gills or the body surface often results in severe mortality. Antibiotic treatment is likely to control the disease. As the disease is triggered by poor biological conditions in hatchery systems, the best control measure is the identification and control of the primary cause. Brown spot disease. Brown spot disease, also known as black spot or shell disease, occurs commonly in juveniles and adult macrobrachium. It can be recognized by the presence of brown to black, ulcerative to raised lesions on the body surface or appendages. This disease occurs only in animals which have developed prior cuticular damage due to other causes, including aggression between prawns. Both adults and larvae have been found to develop melanized brown spot lesions. The only control measure suggested is improvement of culture conditions and correction of nutritional deficiencies. Harvesting. The simplest system, which prawn culturists call batch culture, consists of stocking a pond and allowing the stock to grow until they attain marketable size, after which the whole stock is harvested. For harvesting the stock effectively in this system, the ponds have to be drained. The other techniques involve continuous culture and continuous stocking and harvesting. In continuous culture, the ponds are stocked generally once a year at a comparatively higher rate, and harvesting is done by seines on a continuing basis. After about 5 to 7 months, market-sized prawns are culled at regular intervals. The ponds are never drained. In the continuous stocking and harvesting system, ponds are restocked up to 6 times a year after culling. Some farms try to combine the main features of the different systems. About 5 months after stocking the post-larvae, regular cull harvesting is done until about 8 months, when the pond is drained and the whole remaining stock is harvested. Thanks for watching. Like and share this video. Subscribe this channel to get more informative videos and to become part of a growing scientific community.